Hello, and welcome to our session on building biology overnight with the Gibson SOLA Platform. You can build the DNA you want when you want it. My name is David Weiss. I'm the Director of Corporate Development and Marketing at Telesis Bio. Thank you for joining me, and I'm excited to tell you a bit more about how the Gibson SOLA Platform can enable you to move at the speed of digital biology. The concept of digital biology has been around since the early 2000s, but the definition of the term has evolved significantly from the concept of using digital tools to interpret genomic sequencing data. By turning DNA into code and cells into programmable systems, we're rewriting the rules of biology. With the power of AI, on-demand DNA synthesis, and automation, digital biology doesn't just study life, it builds it. Currently, there are a number of factors that create challenges to enabling the power of digital biology. As I mentioned previously, DNA goes hand in hand with AI and automation to enable digital biology. If we look at some of the markets that are heavily reliant on DNA, you can see that they are growing at an exceptional rate from 13 to 18%. This puts an increased demand on an already strained DNA synthesis infrastructure, as seen by the introduction of variable demand-based pricing by some providers. This doesn't take into account the impact to supply that could be caused by another global health pandemic. The more challenging it is to get your DNA, the more impact it will have on your workflow. Whether we're looking at vaccine development, genome engineering, or antibody discovery, DNA synthesis remains a universal bottleneck to the design, build, test cycle, ultimately slowing down innovation and reducing productivity. Having said that, historically, insourcing DNA synthesis on a broad scale was just not feasible. DNA synthesis has been fraught with challenges. Due to the high level of technical expertise required, the investment needed for very purpose-specific capital expenses, as well as the disposal of toxic reagents, the limited throughput associated with these systems, and the challenges with maintaining high-quality DNA, the only real option for most researchers was to outsource to service providers. At Telesis Bio, we're changing that paradigm by enabling on-demand synthesis powered by a novel enzymatically based, non-toxic approach to DNA synthesis. Our driver with the Gibson SOLA platform is to enable you to make the DNA you want when you want it. By combining the Gibson SOLA platform with Beckman-Coulter automation systems, we are enabling you to eliminate the bottlenecks, boost efficiency, and take better control of your workflow. These benefits result in an accelerated timeline to reliable results with confidence in your biology. By giving you greater transparency into and control over the building of your DNA, you can rapidly iterate on designs that have the benefit of improved characterization and feedback as the DNA you are synthesizing is of a high quality with low variability. This allows you to train your AI models up to 14 times faster than traditional methods to improve these models' predictive powers and reliability. So I've talked a lot about the challenges that exist in the market today and how that impacts current operations, but how does the Gibson SOLA platform allow you to move at the speed of digital biology? When we look at historical service provider-based workflows, the time it took to submit a digital sequence to the time you would receive a sequence-verified construct could take up to 6 weeks, and in some cases, you might get a notice that your sequences couldn't be built after all that time. With the Gibson SOLA workflow, you can design your sequences and synthesize them on demand in one day. This enables you to better plan your downstream experiments in a more controlled fashion, ultimately resulting in a 14-fold increase in time to results. One of the benefits of the Gibson SOLA platform is the fact that all of your reagents are made to stock and kept on site in your facility. These reagents, when combined with a robust design software solution that will analyze your sequences and provide immediate feedback on buildability and optimization, along with a reliable industry-standard automation platform such as the systems provided by Beckman-Coulter, enable you to go from digital sequence to endpoint product in one day. This solution empowers you to build the DNA you need at the scale you need to support your downstream assays regardless of your therapeutic area of focus. Gibson SOLA is a unique take on DNA synthesis. Instead of building a short sequence of DNA base by base as employed by traditional phosphoramidite or enzymatic synthesis, Gibson SOLA uses a pre-qualified short blocks of DNA that can be assembled into most any DNA sequence. This non-toxic process enables on-demand synthesis of high-fidelity, full-length DNA. Let's watch a short video of how Gibson SOLA works.
Enzymatic DNA synthesis, or EDS, has promised to provide a safer, non-toxic alternative to conventional chemical oligonucleotide synthesis. But EDS has been challenging to effectively implement until now. With Telesys Bio's proprietary short oligonucleotide ligation assembly technology, or SOLA, automated high-fidelity and low-cost EDS is possible. SOLA's highly differentiated approach enables rapid construction of synthetic oligonucleotides from digital DNA sequence for a range of life science applications, and enables highly accurate synthesis of full-length genes and beyond. Building DNA with SOLA begins with the selection of short 5-base building blocks from a predefined universal collection. These short overlapping pieces of DNA are stitched together to build any 100 base pair sequence on demand through just a few cycles of hierarchical assembly. SOLA offers inherent error correction so that only perfectly joined sequences are selected and elongated, delivering extremely high fidelity. In addition, because the SOLA input library supports millions of uses, the cost of DNA synthesis drops dramatically. By combining low cost with fast, high-fidelity DNA synthesis, SOLA enables the accurate generation of long DNA, mRNA, and protein, making on-demand desktop printing of biology a reality. Learn more about SOLA by visiting telesysbio.com/SOLA.
As shown in this video, by bringing these short, universal building blocks together, Gibson SOLA only requires 4 cycles of hierarchical assembly to reach the same length that would require 100 cycles using traditional methods. This allows for significantly higher fidelity than other methods, resulting in error rates below 1 in 70,000. When we look at the building process in a bit more detail, we typically break the process up into 3 stages: the pooling, the DNA synthesis, and the gene or construct assembly. Starting with 4 stock plates of universal building blocks, these blocks are pooled based on the sequences of the genes that are being built. Once these custom pools are generated, they go through 4 cycles of hierarchical assembly to generate 100 base pair double-stranded DNA subunits. These subunits are then pooled into wells corresponding to the specific genes that are being built for final construct assembly. This process takes about a day from design to final construct. This process results in uniquely high-quality DNA. Let's take a look at the Gibson SOLA specifications. I'll highlight a few of the most impactful specs. The platform currently supports up to 4 kb of natively synthesized DNA. These sequences can have between 30 to 60% overall GC, but local variation can be as high as 85%. In the next couple of slides, I will talk more about the percent full-length product, yield, and fidelity of the final constructs. I would also like to mention that we've defined these specs to ensure that sequences that fall into our spec ranges will have a very high likelihood of successful builds. But the fact that you have control of your DNA synthesis gives you the option to choose to build outside of our supported specs. While percent full length or yield might be somewhat impacted, you'll still have the opportunity to get your desired sequence. As we focus a bit more on the purity and yield, the Gibson SOLA platform enables greater than 80% full-length DNA, so long as the sequences are within the specified complexity parameters. This is to ensure that you have confidence in the build of your desired sequences. However, if we look at a data set of 80 sequences built across various sizes from 400 base pairs up to about 303 kb and 40 to 60% GC content, you see higher than expected performance. The box and whisker plot shows that the percentage of full-length product for the population is clustering around 95%, with a couple of outliers as low as 80%. On the right, you can see a representative fragment analyzer result from a recent Gibson SOLA run showing a very clean peak. In all cases, we produced full-length product at microgram quantities, analyzed it, and used it for downstream testing. If we further delve into the fidelity, you can see here some data generated using PacBio hi-fi sequencing. Sequencing was performed to an average depth of at least 8,000x, and sequences ranged from 2 kb up to about 7 kb. Error rates were consistently lower than 1 in 70,000 and often exceeded 1 in 100,000. While I touched on it previously, I wanted to specifically highlight one of the additional benefits of the Gibson SOLA platform. By taking control of your DNA synthesis needs, you have the ability to keep your sequence information confidential. There's no risk to your critical IP. We will support 2 versions of the sequence analysis software. One is cloud-based version that will analyze your sequences automatically and provide sequence-specific feedback without review by a Telesys Bio employee, and a fully on-site implementation with the utmost confidentiality for your sequences. Determine your sequence confidentiality needs Submit the sequences you need, iterate often, build your desired sequences on-site with stock universal building blocks. The last specification that I wanted to touch on was length. We currently support constructs from 300 base pairs up to 4 kb. Remember, due to the nature of the hierarchical assembly that is enabled by pre-qualified universal building blocks, you can build any sequence that meets our specifications, including genes up to 4 kb and many outside of our specs. In the next couple of slides, I'll show you some data looking at overall performance, as well as looking at ways to build beyond our stated specifications. Let's look at some fragment analyzer data from various constructs that were synthesized using Gibson SOLA. On the left-hand side of the slide, we see a series of traces evaluating the percent full-length product of sequences ranging from 400 base pairs up to 3.2 kb. In these traces, you can see that the percent full-length ranges from about 92% to greater than 96% full-length. As you can see, we were able to synthesize a broad range of sequence lengths with a high degree of full-length product. On the right-hand side of the slide, we were looking at a single sequence that was synthesized in triplicate. The target size was 365 base pairs, and you can see that with a high degree of reproducibility, the percent full-length product ranging from 96 to 97%. So how can Gibson SOLA enable you to build products larger than 4 kb? Here's an example in which a linearized vector with conserved regions of the expression cassette was paired with a synthesized construct that contained the 3' and 5' UTRs. These fragments were assembled using Gibson assembly approach and then amplified using an ultramer with a poly-A to generate a 9.6 kb linear self-amplifying RNA template. As you can see on the right-hand side of the slide, the construct was successfully synthesized using this combined approach with about 85% full-length product. Finally, I'd like to touch on the current automation approach and the flexibility to scale as necessary. The benefits of collaborating with well-established automation partner like Beckman Coulter is the ability to scale using a modular-based approach. Whether you're interested in a few kb per week, tens of kb per day, or higher throughputs, we can work with you to devise a system that can meet your needs. In addition, none of the hardware that is needed for Gibson SOLA Platform is purpose-built, so you can add on to increase your capacity as needed. As I conclude this talk, I wanted to highlight our fixed offering that uses a defined Beckman Coulter automation solution combined with the Telesis Bio BioXP to enable up to 25 kb of DNA synthesis in about a day. All of the necessary scripts for synthesis have been developed and refined. Start synthesizing your DNA on demand today with confidence. It's as simple as designing your gene build, pooling on the Biomech Echo, synthesizing your subunits in a defined I7 configuration, and assembling your final genes on the BioXP. Maximize your output today. Finally, there are 3 ways to access the Gibson SOLA platform. The fastest way is through the Beckman automation. Use your existing i7 and Echo or bring in a new system. Pre-written scripts are ready, and the Beckman team is prepared to handle your installation and training. The second option is to integrate Gibson SOLA into the automation systems you already have. This approach takes more time as Telesis Bio creates custom scripts to ensure your success. The third way is a funding agreement in which Telesis Bio co-develops custom workflows for your team. This option offers the most significant competitive advantage tailored specifically to your needs. There's never been a time like now where you can take control of your DNA synthesis needs to harness the power of digital biology. Eliminate the bottlenecks to accelerate innovation and build your DNA on demand to iterate as rapidly as your AI model. Have confidence in your success. Thank you. If you have questions, please feel free to reach out to me at my email shown on the slide.